Tonight we explore what happens inside your cells while you rest. The quiet repairs, the slow unwinding of molecular clocks, the ancient mechanisms that govern how long a cell can live. Right now, as you settle in and the room grows still, every cell in your body is running a program that began billions of years ago. It's a program of division, of damage, and of restoration. And it never fully stops, not even while you sleep. In 1961, a cell biologist named Leonard Hayflick was working at the Wistar Institute in Philadelphia. The scientific world at that time believed something that turned out to be wrong. They believed that human cells, if you gave them enough nutrients, could divide forever. A Nobel laureate named Alexis Carrel had championed this idea for decades. Cells were immortal. That was the accepted view. Hayflick proved otherwise. He took fetal fibroblasts, structural cells from developing human tissue, and watched them divide in a controlled culture. They divided 40 times, 50 times, sometimes 60, and then they stopped. Not because they ran out of nutrients, not because of contamination. They simply reached a limit. Adult skin cells did the same thing, but with a tighter budget. They managed only 20 to 40 divisions before entering a permanent state of arrest. The cell was alive but no longer dividing. It had counted its own replications and decided, that's enough. Macfarlane Burnet later gave this observation a name. He called it the Hayflick limit. That name stuck. Across the next 2 hours, you'll hear 100 extended observations about what this limit means and what happens when the body tries to work around it. You'll learn how proteins in your blood can reveal the biological age of 11 different organs. You'll hear about the molecular repair crews that fix your DNA while you sleep. You'll meet a wrinkled, nearly hairless rodent that lives 10 times longer than it should. And through all of it, the tone will stay quiet. The pace will stay slow. There's no rush. The cells have been doing this work for a very long time. Let's begin with the limit itself. Fact 1. The Hayflick limit is not a single number. It varies depending on the type of cell and the tissue it belongs to. Fetal fibroblasts, the cells Hayflick originally studied, divide roughly 40 to 60 times before stopping. These are cells involved in building the structural framework of a developing embryo, laying down extracellular matrix proteins like collagen. Their relatively high division count reflects the enormous construction demands of early life, but even they reach a ceiling. After those 40 to 60 population doublings, they enter a state called replicative senescence. The cell stays metabolically active but will never divide again. Fact 2. Vascular endothelial cells line the inside of your blood vessels. They form a single-cell-thick layer that separates flowing blood from the vessel wall. These cells divide roughly 30 to 50 times over a lifetime. That range reflects the constant physical stress they endure, the push and pull of blood pressure, the exposure to circulating toxins and inflammatory signals. Every heartbeat sends a wave of sheer force across these cells. Their replicative budget has to be large enough to handle decades of vascular repair, but it's still finite. When endothelial cells reach their limit, vascular healing slows. The lining of blood vessels becomes less resilient. Fact 3. Your immune system runs on a tighter clock. CD8 positive T lymphocytes, the cells responsible for hunting down virus-infected cells and destroying them, divide only about 20 to 30 times. That's a narrow window. Each time you fight off an infection, some of those divisions get spent. Chronic viral exposures like cytomegalovirus gradually exhaust the memory T-cell pool over decades. By the time a person reaches old age, many of their immune cells have used up most of their replicative capacity. This is one of the core drivers of what researchers call immunosenescence, the slow weakening of the immune system with age. Fact 4. Keratinocytes are the primary structural cells of your skin. They form the outermost barrier between your body and the outside world, stacking in layers that gradually harden, flatten, and eventually shed. These cells divide 20 to 40 times before reaching their replicative limit. The range depends on several factors: the person's age, their cumulative UV exposure history, and the specific region of skin being measured. Sun-exposed skin on the face or hands tends to have keratinocytes closer to the lower end of that range. Skin on the torso, shielded from light for most of a person's life, tends to preserve more of its replicative capacity. Fact 5. Hepatocytes, the main functional cells of the liver, divide roughly 30 to 40 times. That number is high for a fully differentiated organ. It's part of the reason the liver can regenerate so effectively after injury. If you lose a portion of liver tissue to toxic damage or surgical removal, hepatocytes can re-enter the cell cycle and rebuild. But that rebuilding draws down from a limited account. Each round of regeneration brings the cells closer to their Hayflick limit. In chronic liver disease, where damage and repair cycle repeatedly over years, this limit becomes a real constraint on recovery. Fact 6. Osteoblasts are the cells that build bone. They secrete the mineral matrix that gives your skeleton its rigidity and strength. These cells reach their replicative limit at roughly 30 to 40 population doublings. When osteoblast populations begin to exhaust their capacity, the balance between bone formation and bone resorption shifts. More bone gets broken down than gets built. This is one of the cellular foundations of osteoporosis, a condition where bones become porous and fragile. It also explains why fractures heal more slowly in older adults. The construction crew is running low on workers. Fact 7. Deep in the back of your eye, a thin layer of cells called retinal pigment epithelial cells, or RPE cells, supports the photoreceptors that detect light. These RPE cells divide 36 to 60 times, depending on their starting telomere length and how much oxidative stress they've accumulated over the years. The eye is a particularly oxidative environment. Light itself generates reactive molecules inside the retina. Over decades, that oxidative burden chips away at the RPE cells' replicative reserves. When these cells begin to fail, the photoreceptors they support start to degrade. This process contributes to age-related macular degeneration, one of the leading causes of vision loss in people over 60. Fact 8. Not all cells play by the same rules. Human dental pulp stem cells, found inside the soft tissue of your teeth, can exceed 80 population doublings in controlled laboratory conditions before showing signs of senescence. That's significantly more than most somatic cell types. These cells sit in a protected niche, insulated from many of the environmental stresses that accelerate aging in skin, blood vessels, and immune tissue. Their extended replicative capacity has made them a subject of interest in regenerative medicine, where researchers look for cell populations that can sustain long-term growth without losing their functional properties. Fact 9. And then there are the cells that don't divide at all. The vast majority of neurons in the central nervous system are postmitotic. That means they exited the cell cycle early in development and never re-entered it. Their population doubling count is effectively zero. A neuron born during fetal development is, in most cases, the same neuron you'll carry into old age. It doesn't get replaced. It can't be swapped out for a fresh copy. This is why neurons depend so completely on internal repair mechanisms, on autophagy, on antioxidant defenses, and on the restorative chemistry of sleep. They have no backup plan. What they have is maintenance. Fact 10. The biological clock behind the Hayflick limit has a physical structure. It lives at the ends of your chromosomes in regions called telomeres. Telomeres are made of a repeating six-letter DNA sequence, T, T, A, G, 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 repeated over and over, sometimes thousands of times. They don't encode proteins. They don't carry instructions for building anything. They exist to protect the meaningful genetic information further down the chromosome, the way a plastic cap protects the end of a shoelace. Every time a cell divides, the enzyme that copies DNA polymerase, can't quite reach the very tip of the chromosome. A small segment gets lost. Roughly 50 to 100 base pairs disappear with each division. Fact 11. In some rapidly dividing cell populations, that loss accelerates. Human lymphoblasts, immune cells stimulated to grow quickly in laboratory cultures, can lose up to 120 base pairs per division cycle. Hematopoietic cells undergoing rapid expansion outside the body have been observed losing between 1,000 and 2,000 base pairs of telomeric DNA in just 4 weeks of culture. The loss isn't always gradual. Under conditions of high replicative demand, the telomere clock ticks faster. Fact 12. When telomeres shorten past a critical threshold, the protective protein complex that normally shields them, called the shelter-in complex, can no longer hold on. The exposed chromosome ends look to the cell's surveillance systems like broken DNA. The cell interprets this as a double-strand break, one of the most dangerous forms of genomic damage. A signaling cascade kicks in, driven primarily by the p53 and p21 pathways, and the cell enters irreversible growth arrest. Researchers call this the M1 senescence phase. It's the molecular tripwire beneath the Hayflick limit. Fact 13. Telomere shortening doesn't depend only on cell division. Oxidative stress, the accumulation of reactive oxygen species inside the cell, can damage telomeric DNA directly. A single oxidative lesion at the wrong location can stall the replication fork during cell division. When that happens, the cell can lose up to 1,000 base pairs of telomeric DNA in a single cycle. That's 10 to 20 times the normal rate of attrition. UV radiation, chronic inflammation, and metabolic overload all increase the oxidative burden on telomeres. The clock doesn't just tick. Sometimes it jumps. Fact 14. Certain cell populations partially escape this countdown. Embryonic stem cells and some adult transit-amplifying cells express an enzyme called telomerase. Telomerase is a ribonucleoprotein part RNA, part protein, that adds new copies of the TTAGGG repeat sequence to the ends of telomeres after each division. It effectively resets the clock, restoring what was lost and preserving the chromosome's protective caps. This is how stem cell reservoirs maintain their replicative capacity over years and decades. but most adult somatic cells keep telomerase switched off. For them, the countdown continues with each division, uninterrupted. Fact 15. The replicative lifespan of skin cells can be measurably influenced by UV exposure history. Keratinocytes harvested from sun-exposed areas of the body show shorter telomeres and fewer remaining population doublings compared to keratinocytes from protected skin on the same person. This is not a theoretical prediction. It's a directly measurable difference in the cell's remaining replicative capacity. The environment writes itself into the biology. Every hour of cumulative sun exposure leaves a trace in the telomere lengths of the cells closest to the surface. The Hayflick limit is partly inherited and partly earned. These first 15 observations describe how cells count their own divisions, how telomeres shorten, how different tissues operate on different budgets, and how the environment can accelerate the clock. The next group of facts steps back from individual cells and looks at the whole body, because aging, it turns out, can be read in the blood. Fact 16. In a 2019 study published in Nature Medicine, a team led by Dr. Tony Wiscorey at Stanford measured nearly 3,000 specific proteins in the blood plasma of 4,263 individuals. The participants ranged in age from 18 to 95. By analyzing the patterns across all those proteins, the researchers built what they called a protomic clock, a way of estimating a person's biological age based purely on their blood chemistry. They found that measuring just 373 specific proteins could predict a person's age with a margin of roughly 3 years. 3 years from a blood sample. Fact 17. The composition of blood plasma doesn't change slowly and steadily from youth to old age. When the Stanford team mapped the protein data across their entire cohort, the shift was visible, dramatic color-coded changes rolling across thousands of individuals from young to old. Wiscorey described it in direct terms. They had several thousand individuals. They measured 3,000 proteins in each one, and they could see a dramatic transformation from young people to old people. Not a gradual fade. A clear, readable shift. Fact 18. The protomic clock doesn't just estimate your overall age. A follow-up study in 2024 expanded the method to 44,498 individuals from the UK Biobank. By tracing specific proteins back to their tissues of origin, the research team modeled the biological age of 11 distinct organ systems within each person: the brain, heart, lungs, liver, kidneys, muscles, arteries, pancreas, immune system, intestine, and adipose tissue. Different organs age at different rates inside the same body. Your liver might be biologically younger than your kidneys. Your heart might be older than your brain. The calendar says one age. Your organs say several. Fact 19. The biological age of specific organs carries real predictive weight. Individuals whose brains showed accelerated biological aging, meaning their brain's protomic profile looked older than expected for their chronological age, faced a significantly higher mortality risk. They also showed a drastically increased likelihood of developing Alzheimer's disease within the following decade. This held true regardless of how old the person actually was by the calendar. A 55-year-old with a biologically old brain carried more risk than a 70-year-old with a biologically young one. Fact 20. One of the most important findings in longevity research over the past decade is that aging does not happen at a constant rate. It's nonlinear. The 2019 WIS-Core study identified three distinct protomic waves, periods when the levels of hundreds of blood proteins, which had been relatively stable for years, underwent sudden and coordinated shifts. These waves peaked at approximately ages 34, 60, and 78. During each wave, proteins associated with specific biological processes surged or dropped in ways that could not be explained by simple chronological progression. Fact 21. The protomic wave at age 60 is heavily enriched for proteins associated with cardiovascular disease. Blood vessels, cardiac muscle, and inflammatory signaling pathways all show pronounced shifts during this period. The wave at age 78 carries a different signature. It's heavily enriched for proteins linked to Alzheimer's disease and bone morphogenetic protein signaling. These are not random fluctuations. They reflect coordinated changes in the underlying biology, as if the body is reorganizing itself around new metabolic priorities at each stage. Fact 22. A massive 2024 study published in Nature Aging went even further. Michael Snyder and colleagues at Stanford tracked a longitudinal cohort of 108 healthy volunteers between the ages of 25 and 75. They assessed 135,000 different molecules and microbes, RNA transcripts, proteins, metabolites, lipids, cytokines, and the composition of the gut, skin, and oral microbiomes. The dataset contained 250 billion distinct data points. It was one of the most comprehensive molecular portraits of human aging ever assembled. Fact 23. The Snyder study found that roughly 81% of all the tracked molecules and microbes did not change in a smooth, chronological fashion. Instead, they transformed abruptly, clustering into two intense aging bursts. The first burst averaged at age 44, the second at age 60. These were not gentle curves. They were inflection points at periods where the molecular landscape of the body shifted sharply and quickly before settling into a new baseline. Fact 24. The aging burst in the mid-40s is characterized by large-scale changes in lipid metabolism, cardiovascular disease pathways, and the body's ability to metabolize alcohol and caffeine. The enzymes and proteins that handle these processes undergo sudden, measurable shifts. Initially, the research team suspected this mid-40s burst might be an artifact of perimenopause, a hormonal transition that occurs in women around that age and could skew the data. But the numbers told a different story. Men experience the same dramatic biomolecular shifts in their mid-40s. The burst is not sex-specific. It appears to be a fundamental biological inflection point built into the human aging trajectory. Fact 25. The second aging burst, occurring in the early 60s, shows sharp alterations in immune regulation, carbohydrate metabolism, and kidney function. A secondary wave of cardiovascular disease markers also rises during this period. This helps explain a clinical pattern that doctors have observed for decades: the sudden spike in severe chronic illness during a person's seventh decade. Type 2 diabetes, osteoarthritis, and severe cardiovascular events don't increase smoothly over time. They cluster, they accelerate, and the molecular data now shows why. The body's internal chemistry is reorganizing in ways that make these conditions more likely during this specific window. Fact 26. The wave at age 34, identified in the 2019 study, is less well characterized but still significant. It represents the first major coordinated shift in the plasma proteome after young adulthood. For many people, the biological machinery that maintained relative stability through their 20s begins to change course. Proteins associated with structural tissue maintenance and metabolic regulation shift in ways that reflect the body's transition from peak physiological capacity toward a new, slowly declining baseline. You don't feel it, but the blood shows it. Fact 27. These protomic shifts are not abstract. They correspond to real, measurable changes in organ function. The 11-organ model developed by Wiscore's team in 2024 showed that when a specific organ's protomic age accelerates, meaning its protein signature begins to look older than the person's chronological age, the risk of disease in that organ increases significantly. An accelerated liver signature predicted liver disease. An accelerated kidney signature predicted renal decline. The blood acts as a distributed reporting system, carrying molecular dispatches from every organ in the body. Fact 28. The protomic clock also reveals something about the nature of aging itself. It is not a single process. It is many processes happening in parallel at different rates in different tissues, punctuated by sudden bursts of coordinated change. The body does not age the way a machine wears down uniformly, predictably, part by part. It ages more like a landscape shifting through seasons, long periods of relative stability, then rapid transformation, then a new equilibrium. Fact 29. Recent work at Stanford has also shown that aging affects how the brain handles its own proteins. As the brain ages, the breakdown and clearance of synaptic proteins, the molecules that allow neurons to communicate with each other, slows down. Proteins that should be recycled and replaced begin to accumulate. They aggregate in clusters. Some of these aggregated proteins end up inside microglia, the brain's resident immune cells, where they contribute to chronic low-level inflammation. This slow-motion protein traffic jam is one of the hallmarks of cognitive aging. Fact 30. All of these measurements, the protomic clocks, the organ-specific aging models, the aging bursts, point toward a single observation. The body keeps a detailed biological record of its own age. That record is written in the proteins circulating in the blood, and it can be read. It can even be compared, organ by organ, against the calendar. For the first time in the history of medicine, aging is becoming something that can be precisely measured, not just felt, not just estimated, but quantified down to the level of individual tissues. These first observations describe how cells count their own divisions and how proteins in the blood reflect the age of each organ. The next group of facts looks at what happens when the body attempts to reverse this process, beginning with sleep. Fact 31. During your waking hours, your brain is under constant metabolic pressure. Neurons fire. Synapses transmit signals. Cognitive processing generates heat, consumes oxygen, and produces waste. One of the most significant byproducts of all this activity is the generation of reactive oxygen species, highly reactive molecules that can damage cellular structures, including DNA. Over the course of a single waking day, this oxidative stress accumulates inside neurons and leads to the formation of double-strand DNA breaks. These are not small nicks or superficial lesions. A double-strand break means both strands of the DNA double helix have been completely severed. Fact 32. Double-strand breaks are widely considered the most dangerous form of genomic damage a cell can sustain. When both strands of the helix are cut, the cell risks catastrophic consequences, chromosomal rearrangements, loss of genetic information, or rapid cell death. In most cell types, a double-strand break triggers an emergency repair response. But neurons face a unique challenge. They are postmitotic. They cannot divide to replace a damaged neighbor. They cannot dilute the damage through fresh copies. Every neuron must repair its own DNA or live with the consequences. Fact 33. The accumulation of double-strand breaks during waking hours creates a form of cellular pressure. The breaks build up steadily as the day progresses. This is not a theoretical concern. It has been directly observed in experimental models. The longer the period of sustained wakefulness, the greater the number of DNA lesions within neuronal nuclei. This accumulating damage is one of the core biological drivers behind the sensation of tiredness. Your body doesn't just want to sleep. At the molecular level, your neurons need to sleep to access the repair window that only deep rest provides. Fact 34. Sleep creates a different chemical environment inside the brain. During slow-wave sleep, the deep, restorative stage known as NREM N3, neuronal firing rates drop dramatically. The electroencephalogram shows large, slow delta waves oscillating between 1 and 4 cycles per second. These synchronized, high-voltage waves represent a state of coordinated neural quiet. In this electrophysiologically calm state, the production of reactive oxygen species falls sharply. The conditions become favorable for repair. Fact 35. The repair process begins with a sensor. An enzyme called PARP1, poly-ADP-ribose polymerase 1, acts as a highly sensitive detector of DNA damage. During waking hours, as double-strand breaks accumulate, PARP1 binds to the damaged sites and undergoes a chemical modification called auto-PA-relation. This modification is itself a signal. As PARP1 activity accumulates throughout the day, it acts as a molecular gauge of cellular exhaustion. Research has shown that this buildup of PARP1 activity directly promotes the onset of sleep. The enzyme doesn't just detect damage. It helps push the brain toward the state in which that damage can be fixed. Fact 36. Once sleep begins and the brain enters slow-wave stages, a second molecular player arrives at the scene. ATM kinase, ataxia-telangiectasia mutated kinase, is recruited to the sites of double-strand breaks. ATM's role is to signal the extent of the damage and to pause any active gene transcription near the break site. This pause is essential. If the cell tried to read genes from a broken stretch of DNA, the results could be garbled or harmful. ATM creates a quiet zone around the damage, clearing the way for the repair machinery to work without interference. Fact 37. The actual rejoining of severed DNA strands is carried out by a system called non-homologous end joining (NHEJ). The first step involves a pair of proteins called Q70 and Q80, which form a ring-shaped structure that slides onto the broken DNA ends. This Q heterodimer acts like a molecular clamp, holding the severed ends in place and preventing them from being degraded further. It's a stabilizing grip, precise, protective, and essential for what comes next. Fact 38. Once Q70 and Q80 have secured the broken ends, they recruit the catalytic subunit of DNA-dependent protein kinase, DNAP-KCS. Together with the XRCC4 and DNA ligase IV complex, this machinery seals the break. The severed strands are rejoined. The integrity of the chromosome is restored. This entire process, detection by PARP1, signaling by ATM, clamping by Cu70/80, and ligation by DNAPKCS and ligase IV, unfolds during the hours of deep sleep. By the time the brain wakes, the DNA damage counter has been partially reset, ready for another day of waking activity. Fact 39. The repair work leaves visible traces. When a double-strand break occurs, the histone proteins near the break site get modified. Specifically, a histone variant called H2AX gets phosphorylated, producing a marker known as gamma H2AX. Another protein, 53BP1, also accumulates at damaged sites. These markers form what researchers call repair foci, small bright spots visible under a fluorescence microscope. Studies have shown that these foci accumulate during wakefulness and resolve during sleep. You can literally watch the neurons mend themselves under the microscope as the animal rests. Fact 40. While this intracellular repair is happening, a second cleaning system operates on a larger scale. The brain has no traditional lymphatic network, no dedicated drainage system like the rest of the body uses to clear waste from tissues. Instead, it relies on a specialized pseudolymphatic plumbing system discovered in 2012 by Macon Nettergaard and her colleagues. They called it the glymphatic system, a name that combines glia with lymphatic, because it depends heavily on glial cells to function. Fact 41. The glymphatic system uses perivascular spaces, channels surrounding the brain's blood vessels, to flush metabolic waste from the central nervous system. Cerebrospinal fluid flows into the brain along the outer walls of arteries, sweeps through the spaces between neurons, collects metabolic byproducts, and drains out along the walls of veins. The waste is then directed out of the brain entirely, through meningeal lymphatic vessels that empty into the deep cervical lymph nodes in the neck. It's a washing cycle, and it runs most efficiently when you're asleep. Fact 42. During NREM sleep, a specific neurochemical shift activates the glymphatic system. The locus coeruleus, a small brainstem nucleus that produces norepinephrine, reduces its activity. Norepinephrine levels drop. This chemical quieting acts as a master switch. In response to the lower norepinephrine levels, astrocytes, the star-shaped glial cells that wrap around blood vessels throughout the brain, physically shrink. Their cellular volume decreases, and the interstitial spaces between brain cells expand. The expansion is not subtle. The brain's interstitial fluid volume fraction increases by 40 to 60% during NREM sleep. Fact 43. That expansion is what makes the washing cycle possible. When the interstitial spaces widen, the resistance to fluid flow drops dramatically. Cerebrospinal fluid can move through the brain tissue with far less friction. It travels from periarterial spaces deep into the brain's tissue, collecting soluble waste products, including amyloid beta and tau proteins, two molecules closely associated with Alzheimer's disease. The flow is driven in part by the pulsing of arteries with each heartbeat, creating a gentle hydraulic push that propels the fluid forward through the expanded channels. Fact 44. The exchange of cerebrospinal fluid and interstitial fluid depends on a specific molecular gateway. Aquaporin-4 water channels, densely clustered on the vascular end feet of astrocytes, act as the primary portals through which fluid enters and exits the brain tissue. These channels are not distributed randomly. They are polarized, concentrated specifically at the points where astrocytes contact blood vessels. Without functional aquaporin 4 channels, glymphatic clearance drops significantly. The channels are the doors. Sleep opens them. Fact 45. The efficiency of glymphatic waste clearance during sleep has been measured directly. Experimental models show that the clearance rate of soluble amyloid beta is up to twofold faster during natural sleep compared to wakefulness. That means the brain clears this particular waste product at 200% of its waking rate during deep sleep. For a molecule linked to the formation of Alzheimer's plaques, this difference in clearance speed matters enormously over the decades of a human lifetime. Fact 46. The glymphatic system slows down with age. In healthy 30-year-old subjects, the half-life of amyloid beta clearance, the time it takes for the brain to remove half of the accumulated amyloid, is approximately 3.8 hours. In 80-year-old subjects, that half-life extends to roughly 9.4 hours. The waste removal system doesn't stop entirely, but it becomes sluggish. The same brain produces similar amounts of amyloid beta at 30 and at 80. But at 80, the cleaning crew takes more than twice as long to remove it. The waste accumulates. Over years, this slow buildup contributes to the plaques that define neurodegenerative disease. Fact 47. Sleep and DNA repair are not merely correlated. They are mechanically linked. The same slow-wave oscillations that create the calm neural environment for NHEJ repair also generate the norepinephrine withdrawal that activates glymphatic clearance. Two different repair systems, one operating inside the nucleus of individual neurons, the other flushing waste through the entire brain, are both gated by the same sleep stage. Deep NREM sleep is the key that opens both locks simultaneously. Fact 48. The relationship between sleep and DNA repair has been observed across species. In fruit flies, researchers tracked double-strand break markers in neurons during wake and sleep cycles. The pattern was consistent. Damage accumulated during active waking periods. Repair occurred during rest. When the flies were sleep-deprived, kept awake beyond their normal cycle, the DNA damage markers continued to rise without resolution. The repair window requires actual sleep, not just reduced activity. Rest without sleep is not enough. Fact 49. In mice, similar observations hold. Sleep-deprived mice show elevated levels of gamma-H2AX foci in their neurons, the phosphorylated histone markers that flag double-strand breaks. When the mice are allowed to sleep normally, those foci diminish. The repair machinery engages during sleep, processes the accumulated breaks, and restores genomic integrity before the next waking period. Chronic sleep deprivation in these models leads to a progressive buildup of unresolved DNA damage. The repair debt grows over time. Fact 50. So the picture looks like this. During the day, your brain works. Neurons fire, synapses transmit, and thoughts take shape. That activity produces oxidative waste and inflicts DNA damage, hundreds of tiny breaks accumulating across billions of neurons. Then night falls, core body temperature drops, neuronal firing slows. The brain descends into slow-wave sleep. PARP1 signals the onset. ATM marks the damage. Q70 and Q80 clamp the broken ends. DNAPKCS and ligase IV seal the breaks. At the same time, astrocytes shrink, interstitial spaces expand, and cerebrospinal fluid washes through the tissue, carrying away the amyloid debris of the day. By morning, the damage has been partially reversed. the cycle begins again. These facts describe the quiet chemistry of human sleep, the temperature shifts, the hormonal signals, the slow repair of damaged DNA. But there is more to the nightly restoration than repair enzymes and waste clearance. There are also the hormones, the chemical messengers that regulate how deeply you sleep and how well your cells age. Fact 51. Falling asleep requires a specific physical trigger. Your core body temperature must drop by 1 to 3 degrees Fahrenheit, roughly half a degree to 1.5 degrees Celsius, before the brain can descend into the deepest stages of slow-wave sleep. This is controlled by the suprachiasmatic nucleus, the brain's master circadian clock. As evening approaches, the body begins shedding heat through a process called distal vasodilation. Blood vessels in the hands and feet dilate, flushing warm blood away from the core and toward the extremities. Your core cools, your fingertips warm, and that temperature drop is what tells the brain it's time to go deep. Fact 52. The temperature nadir, the lowest point of core body temperature during the night, is not just a side effect of resting. It's an active trigger. The suprachiasmatic nucleus coordinates the timing of this drop to coincide with the onset of the deepest NREM sleep stages. If the drop is prevented, if the room is too warm, or if the body's thermoregulatory system is disrupted, the brain has difficulty reaching the slow-wave stages where the most intensive repair occurs. The relationship between temperature and deep sleep is not incidental, it's causal. Fact 53. Alongside the temperature drop, the pineal gland begins secreting melatonin. Most people think of melatonin as a sleep hormone, something that makes you drowsy. That's partly true, but it misses the larger picture. Melatonin functions as what endocrinologists call a transducer. It converts the external signal of light and darkness into an internal chemical calendar. As daylight fades and darkness sets in, melatonin levels rise. This rise doesn't just promote sleepiness. It communicates seasonal information to tissues throughout the body, influencing gene expression, immune function, and cellular repair processes on timescales that span months. Fact 54. Melatonin has a direct and measurable effect on telomere maintenance. In controlled laboratory models, the administration of melatonin resulted in a 3.9-fold increase in telomerase activity within aortic tissue. In liver tissue, the increase was even greater, 5.13-fold. Telomerase, as we discussed earlier, is the enzyme that adds new TTAGGG repeats to telomere ends, counteracting the shortening that occurs with each cell division. Melatonin doesn't just help you fall asleep. It activates the enzyme that resets part of the cellular aging clock. Fact 55. Melatonin also acts as an antioxidant, and a potent one. It scavenges free radicals directly, neutralizing the reactive oxygen species that would otherwise damage telomeric DNA. It preserves mitochondrial oxygen metabolism, reducing the oxidative burden on the cell from within. This dual action, stimulating telomerase while simultaneously reducing the oxidative damage that accelerates telomere shortening, makes melatonin one of the most directly relevant sleep-associated molecules in the biology of cellular aging. Fact 56. The flip side of melatonin's protective effects is cortisol. Chronic sleep deprivation elevates cortisol levels and hyperactivates the hypothalamic-pituitary-adrenal axis, the hormonal stress system that mobilizes the body's resources during perceived threats. When cortisol stays elevated for prolonged periods, it generates systemic oxidative stress. That oxidative stress accelerates the shortening of immune cell telomeres, the very cells that defend you against infection and disease. Chronic sleep loss doesn't just make you feel tired. It ages your immune system at the molecular level. Fact 57. The deepest stages of sleep also regulate the pulsatile release of growth hormone from the pituitary gland. Growth hormone secretion peaks during slow-wave sleep, with the largest bursts occurring in the first half of the night. This nightly pulse of growth hormone triggers the liver to produce insulin-like growth factor 1, or IGF-1. Together, growth hormone and IGF-1 support protein synthesis, tissue repair, and cellular maintenance across the body. Muscles recover. Bones remodel. Damaged tissues rebuild. The sleep window is when much of this work gets done. Fact 58. The relationship between IGF-1 and longevity is paradoxical. On a systemic level, reduced IGF-1 signaling is one of the most consistently documented pathways for extending lifespan across species. Ames dwarf mice, which have severe growth hormone deficiencies, live significantly longer than normal mice. Their cells experience lower metabolic stress and reduced activation of the mTOR nutrient-sensing pathway. Less IGF-1, systemically speaking, appears to mean a longer life. Fact 59. But inside the brain, the story inverts. Localized IGF-1 acts as a critical neuroprotector. It signals through the Pi3K-ACT pathway to maintain neuronal mitochondria and support synaptic plasticity, the brain's ability to strengthen and reorganize its connections. IGF-1 also inhibits a form of neurodegeneration driven by tau protein accumulation. It does this by downregulating two specific enzymes, HDAC1 and HDAC3, that would otherwise promote the toxic aggregation of tau. The same molecule that shortens lifespan when elevated throughout the body protects the brain when present locally. Fact 60. This paradox highlights a theme that runs through much of aging biology. The body is not optimizing for a single outcome. It balances competing demands, growth versus maintenance, systemic longevity versus local neuroprotection, repair versus replication. Sleep is one of the few states in which many of these competing demands can be addressed simultaneously. Hormones pulse, DNA gets repaired, waste gets cleared, telomeres get partially maintained. No single mechanism does all the work. The body coordinates dozens of processes in parallel, gated by the same transition into deep sleep. Fact 61. Light exposure during the day plays a direct role in the quality of the hormonal cascade at night. The suprachiasmatic nucleus receives input from specialized photoreceptors in the retina, cells called intrinsically photosensitive retinal ganglion cells, that detect the intensity and spectral quality of ambient light. Bright, blue-shifted light during the morning hours strengthens the circadian signal and promotes a more robust melatonin pulse at night. Dim, warm light in the evening supports the transition. The hormonal architecture of sleep is built during the waking hours, shaped by light exposure patterns that most people rarely think about. Fact 62. Even the skin responds to light at the cellular level. Melanocytes, the pigment-producing cells in the epidermis, activate genetic programs in response to UV radiation. These programs don't just produce melanin for skin color. They trigger DNA repair pathways, stress response genes, and signaling cascades that communicate with surrounding cells. The boundary between the body's internal environment and the external world is thinner than most people assume. Light doesn't just enter through the eyes. It reaches the skin and alters gene expression in the cells closest to the surface. Fact 63. The hormonal regulation of sleep is also deeply tied to the immune system. During deep sleep, the balance between pro-inflammatory and anti-inflammatory cytokines shifts. Levels of interleukin-6, a key inflammatory marker, follow a circadian rhythm that peaks during sleep, but in a controlled, pulsatile pattern. Chronic sleep disruption flattens this rhythm, leading to sustained low-grade inflammation. This kind of persistent, unresolved inflammation, sometimes called inflammaging, accelerates telomere shortening, promotes cellular senescence, and increases the biological age of multiple organ systems simultaneously. Fact 64. The temperature drop, the melatonin pulse, the growth hormone surge, the cortisol suppression, the immune rebalancing, all of these happen in sequence, layered on top of one another across the stages of a single night's sleep. The first half of the night tends to be dominated by deep slow-wave sleep and the associated growth hormone release. The second half shifts toward lighter sleep stages, REM sleep, and different hormonal patterns. The body moves through these stages in roughly 90-minute cycles, each cycle recalibrating a different set of molecular parameters. Fact 65. And none of this machinery works well without consistency. The circadian system is built on expectation. It anticipates the timing of light, meals, activity, and rest based on prior patterns. When those patterns are disrupted by shift work, jet lag, late-night light exposure, or irregular sleep schedules, the hormonal cascade loses its coordination. Melatonin arrives too late. Cortisol lingers too long. Growth hormone pulses weaken. The DNA repair window narrows. The molecular repair work still happens, but it happens less efficiently. The cells accumulate a small but measurable debt with each disrupted night. These facts describe the quiet chemistry of human sleep, the temperature shifts, the hormonal signals, the slow repair of damaged DNA. The next group of facts looks at animals that have found other solutions to the problem of cellular aging. Fact 66. The naked mole rat is a rodent. It's about the size of a mouse. It lives underground in East Africa, in tunnels it digs through dry, hard soil. It's wrinkled, nearly hairless, and looks, by most human standards, deeply unusual. It also lives more than 30 years. That's roughly 10 times the lifespan of a similarly sized house mouse. A house mouse typically lives 2 to 3 years. The naked mole rat lives past 30, sometimes past 40, and it does so with almost no cancer. Fact 67. The naked mole rat's resistance to cancer is not a matter of luck or diet. It's built into the cellular machinery. When naked mole rat cells begin to crowd together in tissue, approaching the density at which tumors might start to form, they trigger a growth arrest response far earlier and far more strongly than human or mouse cells do. Researchers call this early contact inhibition. In mice and humans, cells use a gene called P27 to enforce regular contact inhibition, slowing growth when the tissue gets dense. Naked mole rat cells use a two-tiered defense. They activate the P16 gene pathway at a much lower cell density, halting proliferation before the tissue even approaches a dangerous threshold. Fact 68. The P16 response in naked mole rats is mechanically driven by a molecule called high molecular mass hyaluronan, HMMH. This is a very large sugar molecule, part of the extracellular matrix that fills the spaces between cells. Naked mole rats produce an unusually abundant supply of this molecule. When HMMHA interacts with CD44 receptors on the cell surface, it triggers the P16 pathway. The cell receives a clear, early signal: stop dividing. This mechanism acts as a preventive barrier against tumor formation, catching potentially dangerous growth before it gains momentum. Fact 69. But the naked mole rat's most remarkable adaptation for longevity involves a protein called CGAS, cyclic GMP-AMP synthase. In humans and mice, CGAS serves primarily as a sensor for foreign or misplaced DNA. If viral DNA appears in the cell's cytoplasm, or if damaged DNA leaks out of the nucleus, CGAS detects it and triggers an inflammatory immune response through the STING pathway. That immune response helps fight infections but comes at a cost. When human CGAS moves into the nucleus after DNA damage, it actually inhibits one of the most accurate forms of DNA repair, homologous recombination. In humans, the very protein that detects damage also interferes with fixing it. Fact 70. The naked mole rat has rewritten this system. Through evolutionary selection, it has acquired 4 specific amino acid substitutions in the C-terminal domain of its CGAS protein. These substitutions occur at positions 463, 511, 527, and 530, serine, glutamate, tyrosine, and threonine, respectively. 4 changes, 4 amino acids out of hundreds. That's all it took. Fact 71. Those 4 substitutions fundamentally change how CGAS behaves after DNA damage. In humans, CGAS gets tagged with a molecular label called K48-linked ubiquitin by an enzyme called TRIM41. That ubiquitin tag is the cell's signal for degradation. It marks CGAS for destruction. After the tag is attached, a protein called P97, a segregase, physically extracts CGAS from the chromatin and sends it to be broken down. The human version of CGAS is rapidly cleared from the site of damage. In the naked mole rat, those 4 amino acid changes reduce the ubiquitination by TRIM41 and weaken the interaction with P97. The result is that naked mole rat CGAS doesn't get degraded quickly. It persists on the chromatin much longer than the human version. Fact 72. That persistence matters enormously. Instead of inhibiting DNA repair the way human CGAS does, the naked mole rat's modified CGAS acts as a molecular scaffold. It enhances the interaction between 2 proteins critical for homologous recombination, FANCI and RAD50. By holding these proteins together at the damage site for longer, the mole rat version of CGAS supercharges the repair process. Double-strand breaks that would push human cells toward senescence or malignant mutation get seamlessly repaired in the naked mole rat. Fact 73. Researchers tested whether these 4 amino acid changes were sufficient to explain the longevity benefit. They engineered fruit flies, Drosophila, to carry CGAS with the same 4 mole rat substitutions. The modified flies showed improved DNA repair efficiency, reduced markers of cellular senescence, and an extended lifespan compared to flies with the normal version of CGAS. 4 amino acid changes, a longer life. The experiment demonstrated that these specific residues function as a genetic switch for longevity. Fact 74. The naked mole rat's approach to cellular aging is fundamentally different from the human approach. Human cells rely heavily on telomere attrition to enforce replicative limits. When telomeres get short, the cell stops dividing. The naked mole rat uses a different strategy. It maintains genomic stability through superior DNA repair, early growth arrest, and a modified immune sensor that enhances rather than inhibits the repair process. The telomere clock still operates, but it runs alongside robust backup systems that prevent the accumulation of damaged, dysfunctional cells. Fact 75. Under strong stressors, high levels of DNA damage, oxidative stress, or oncogenic signals, naked mole rat cells bias heavily toward one of three outcomes. They either arrest tightly, preventing any further division. They repair the damage with high fidelity through homologous recombination, or they eliminate themselves entirely through programmed cell death. What they almost never do is linger in a damaged, partially functional state, secreting inflammatory signals and corrupting their neighbors. This refusal to tolerate intermediate dysfunction is central to the species' extraordinary health span. Fact 76. The naked mole rat also maintains stable protein quality well into its third decade of life. Unlike mice, which show significant protein aggregation and loss of protostases within 2 to 3 years, naked mole rats preserve resilient protein folding and membrane integrity far longer. Their ribosomes, the cellular machines that build proteins, show increased translational fidelity compared to mouse ribosomes. They make fewer errors when reading genetic instructions. The proteins they produce are more likely to fold correctly and function as intended. Fact 77. The environment the naked mole rat lives in is itself hostile to most mammals. Their underground tunnels contain low oxygen levels and high concentrations of carbon dioxide and ammonia. Most rodents would struggle to survive in these conditions. The naked mole rat thrives. Its cells have adapted to function efficiently under metabolic conditions that would stress and damage the cells of surface-dwelling animals. In some sense, the species evolved its longevity mechanisms in response to an environment that demanded extraordinary cellular resilience. Fact 78. The naked mole rat is not the only long-lived rodent. The Brandt's bat, another small mammal with a body mass comparable to a mouse, can live over 40 years. Bowhead whales live over 200 years. Greenland sharks may exceed 400. Each of these species has evolved distinct molecular strategies for managing DNA damage, cellular senescence, and protein quality over extremely long timescales. The naked mole rat is simply the most studied example of a broader biological principle. Longevity is not a fixed property of body size. It's a function of cellular engineering. Fact 79. What makes the naked mole rat especially relevant to human aging research is the specificity of its adaptations. The 4 amino acid changes in CGAS are not vague, system-wide modifications. They are precise, targeted alterations in a single protein that change one biochemical interaction, the interaction between CGAS, TRIM41, and P97. This level of molecular precision suggests that similar targeted interventions might eventually be applicable to human cells. Not tomorrow, not easily, but the path from observation to application begins with understanding exactly which molecular switches matter. Fact 80. It's worth pausing here to notice how different the mole rat's strategy is from the human body's nightly repair routine. Humans depend on sleep, on temperature drops, melatonin, slow-wave oscillations, to access a narrow window of DNA repair and waste clearance. The naked mole rat doesn't rely on the same narrow window. Its cells carry built-in, always available repair enhancements that operate continuously. The human approach is scheduled maintenance. The mole rat's approach is structural reinforcement. Both work, but they represent fundamentally different solutions to the same problem: how to keep a genome intact across a long life. The next group of facts returns to human biology and asks a different question. If aging is written in the blood, in proteins, in hormones, in molecular clocks, can those signals be rewritten? Fact 81. The idea that blood carries aging signals and rejuvenating ones has been tested directly through a surgical technique called parabiosis. In heterochronic parabiosis, researchers surgically joined the circulatory systems of a young mouse and an old mouse. Their blood mixes. Factors present in the young mouse's blood reach the old mouse's tissues, and vice versa. The results, first reported in detail by teams at Stanford and Harvard, were striking. The old mice showed reactivation of dormant neural stem cells, reduced brain inflammation, and improved memory encoding. Fact 82. The effects were not limited to the brain. Old mice connected to young blood supplies showed improvements in muscle repair, liver regeneration, and cardiac function. The rejuvenating factors worked systemically. They traveled through the blood plasma and affected multiple organs at once. The aging process, at least in mice, turned out to be more plastic than anyone had assumed. It could be partially reversed by exposure to the right circulating molecules. Fact 83. The reverse experiment was equally telling. When young mice were exposed to blood from old mice, they showed accelerated cellular decline. Specific pro-aging factors, including a molecule called VCAM1 and various inflammatory cytokines, appeared to drive the deterioration. Aging isn't just the absence of youth. Old blood actively carries molecules that push tissues toward decline. The blood is not neutral. It is an active participant in the aging process, for better and for worse. Fact 84. Wiscore described the implications in clear terms. They could give factors from a young organism and ask whether that would change the age of the brain. And that's indeed what it did. The protomic age of the older animals' brains shifted measurably in response to young blood factors. The brain didn't become young again, but it became measurably younger than it had been. Fact 85. Exercise produces many of the same effects without surgery. Intense muscle contraction and cardiovascular exertion stimulate the release of specific molecules into the bloodstream, molecules that researchers have termed exerkines and myokines. Two of the most studied are irisin and clusterin. These molecules are produced by active muscle tissue, enter the bloodstream, and cross the blood-brain barrier. Once inside the brain, they promote tissue recovery, stimulate the growth of new blood vessels, and support the formation of new neurons in the hippocampus, the brain region most closely associated with memory. Fact 86. The parallel between exercise-derived molecules and young blood factors is not coincidental. Both pathways result in similar downstream effects: reduced neuroinflammation, enhanced neurogenesis, improved cognitive function, and measurable shifts in the protomic age of multiple organs. Exercise provides a natural, repeatable mechanism for flooding the bloodstream with rejuvenating signals. No surgery required. No young mouse involved. Just sustained physical effort, repeated over weeks and months. Fact 87. These rejuvenating factors don't act locally. Irisin produced by a contracting quadriceps muscle doesn't just benefit the leg. It travels through the plasma to the brain, the liver, the heart, and the kidneys. Clusterin does the same. The systemic nature of these molecules means that regular exercise affects the biological age of the entire body simultaneously, directly echoing the multi-organ protomic shifts identified in Wiscore's blood protein studies. Fact 88. The shift from observing aging to intervening in aging represents one of the most significant transitions in modern biology. For most of human history, aging was understood as inevitable, irreversible, and fundamentally beyond the reach of medicine. The protomic clocks, the parabiosis experiments, and the exercise studies have collectively moved the field into new territory. Aging is still inevitable, but specific aspects of it, the protomic age of individual organs, the inflammatory burden of senescent cells, the rate of telomere attrition, appear to be modifiable. Fact 89. One of the key insights from the parabiosis work is that the factors responsible for rejuvenation are specific, identifiable proteins, not vague essences of youth. They can be isolated, measured, and potentially synthesized. Research teams at Stanford and elsewhere are working to identify the precise molecules in young blood plasma that drive the beneficial effects, with the goal of developing targeted therapies that could deliver the same benefits without the need for a direct blood connection. Fact 90. The connection between exercise, young blood, and organ aging reinforces a broader principle. The body's aging trajectory is not fixed at birth. It's continuously influenced by circulating factors, molecules that rise and fall in response to activity, rest, diet, stress, and hormonal rhythms. The protomic clock is not a countdown timer. It's more like a speedometer. Its reading reflects what the body is currently experiencing, and it can accelerate or decelerate in response to changing conditions. Fact 91. For the listener resting quietly tonight, this means something practical. Sleep repairs DNA and clears metabolic waste. Exercise floods the bloodstream with molecules that reduce the biological age of multiple organs. Melatonin activates telomerase and protects telomeres from oxidative damage. These are not future interventions. They are not experimental therapies. They are processes happening inside your body right now, or that happened earlier today, shaped by the choices you made about when to sleep, how to move, and what light to expose yourself to. Fact 92. The distance between a parabiosis experiment in a mouse and a practical intervention in a human is still large, but the distance is shrinking. Each year, more of the specific molecular players are identified. Each year, the protomic models become more precise. Each year, the list of modifiable aging factors grows longer. The field is moving from description to intervention, slowly, carefully, and with the kind of precision that decades of cellular biology have made possible. The final group of facts looks at the tools that researchers are building. Tools designed not to replace the body's repair systems, but to amplify them. Tools that work with the same molecular logic the body already uses. Fact 93. CRISPR-Cas9 gene editing has given researchers the ability to modify specific genes in living cells with a precision that was unthinkable 20 years ago. In the context of aging, CRISPR is being used to study and manipulate the genes that control cellular senescence, telomerase expression, DNA repair efficiency, and immune cell behavior. Researchers can now edit immune cells, T cells, natural killer cells, to enhance their ability to recognize and destroy specific targets. This includes cancer cells that have evaded the immune system by exploiting the very mechanisms of cellular senescence. Fact 94. mRNA technology, which reached public awareness through its use in vaccine development, has broader applications in cellular programming. Synthetic mRNA transcripts can be delivered to cells to temporarily produce specific proteins. In aging research, this approach is being explored as a way to boost the expression of repair enzymes, telomerase, or anti-inflammatory molecules in targeted tissues without permanently altering the genome. The instruction is temporary. The cell reads it, produces the protein, and the mRNA degrades. It's a set of instructions, not a permanent rewrite. Fact 95. One of the most heavily funded areas of longevity research involves a class of drugs called senolytics. The name comes from senescence and lytic, meaning to destroy. Senolytics are pharmacological compounds designed to selectively eliminate senescent cells. These are cells that have reached their Hayflick limit or suffered irreparable DNA damage, but haven't undergone programmed cell death. Instead, they linger in the tissue. They remain metabolically active. And they secrete a toxic mixture of inflammatory cytokines, proteases, and growth factors that researchers call the senescence-associated secretory phenotype, or SASP. Fact 96. Senescent cells are sometimes called zombie cells, alive but dysfunctional, secreting signals that accelerate the aging of surrounding healthy tissue. The SASP includes molecules like interleukin-6 and interleukin-1 alpha, both potent inflammatory signals. Over time, the accumulation of senescent cells drives chronic, low-grade inflammation throughout the body. This localized, persistent inflammation contributes to a wide range of age-related conditions: osteoarthritis, type 2 diabetes, cardiovascular disease, and general frailty. The cells that have stopped dividing don't die quietly. They corrode their neighborhood. Fact 97. The primary senolytic combination currently under clinical investigation is dasatinib and quercetin. Often abbreviated D plus Q, dasatinib is a targeted tyrosine kinase inhibitor originally developed as a chemotherapy agent for leukemia. Quercetin is a naturally occurring plant flavonoid found in foods like onions, apples, and green tea. Together, they target the anti-apoptotic survival networks that senescent cells rely on to stay alive despite their own toxic environment. Dasatinib blocks specific tyrosine kinases, including ephrin dependence receptors, that senescent cells use for survival. Quercetin inhibits Pi3K and AKT pathways, reducing the secretion of SASP factors. Fact 98. Because senescent cells take weeks to reform after being cleared, senolytics don't need to be taken daily. They're administered in what researchers call hit-and-run regimens, brief, intermittent treatments that clear the existing burden of senescent cells, followed by weeks or months of rest before the next course. Phase 2 clinical trials have demonstrated that intermittent treatments with dasatinib and quercetin can safely clear senescent cell burdens in humans. improving physical function and increasing bone mineral density in elderly postmenopausal women with osteoporosis. Pilot studies like the Stamina trial are evaluating whether clearing senescent cells from the central nervous system can improve cerebral blood flow and slow the progression of mild cognitive impairment toward Alzheimer's disease. Fact 99. Alongside senolytics, two other molecules are receiving sustained attention in longevity research. NAD+, nicotinamide adenine dinucleotide, is a coenzyme essential for sirtuin activation and mitochondrial energy transfer. As cells age, NAD+ levels decline, reducing the cell's ability to maintain its energy supply and activate key repair pathways. NAD+ precursors, supplements that the body can convert into active NAD+, are being studied for their ability to restore these declining levels. Rapamycin, originally derived from a soil bacterium on Easter Island, inhibits the mTOR nutrient-sensing pathway. By suppressing mTOR, rapamycin pushes cells toward a state of internal maintenance and autophagic repair rather than rapid, error-prone division. Fact 100. All of these interventions, CRISPR, mRNA delivery, senolytics, NAD+, rapamycin, share a common design logic. They don't replace the body's repair systems. They amplify them. They work with the molecular machinery that already exists, the same DNA repair enzymes that activate during slow-wave sleep, the same telomerase that melatonin stimulates, the same immune clearance pathways that exercise enhances. The goal is not to build something the body doesn't have. The goal is to make what it already has work better, last longer, and reach further. And with that, the 100th observation comes to rest. Right now, as you lie here and the room is quiet, repair enzymes are moving along strands of your DNA. PARP1 is sensing. Q70 and Q80 are clamping. Ligase IV is sealing. In the spaces between your neurons, cerebrospinal fluid is flowing slowly, steadily, carrying away the molecular residue of the day. Melatonin is circulating through your blood, reaching tissues you'll never see, activating telomerase in cells you'll never feel. Your core temperature has dropped. Your cortisol is low. The conditions are right. This is not a passive state. Sleep is active repair. It is the body's most ancient form of maintenance, refined across billions of years of cellular life. When we step back far enough past the Hayflick limit, past the protomic clocks, past the naked mole rat and its 4 amino acid changes, we arrive at a very old story. Cells have been dividing since life began, and for almost as long they have been repairing themselves. The first single-celled organisms in the ancient oceans faced the same fundamental problem that your neurons face tonight: how to fix what the day has broken, how to clear what has accumulated, how to restore what has been depleted. The mechanisms have grown more complex. The organisms have grown larger. The repair systems have been elaborated into cascades of enzymes and hormones and circadian rhythms. But the core logic is the same: divide, damage, repair, rest. The Hayflick limit is not a flaw. It's a boundary that prevents unchecked growth, the kind of growth that becomes cancer. Telomere shortening is not a design error. It's a counting mechanism that ensures cells know when to stop. The protomic waves at 44 and 60 are not breakdowns. They're reorganizations, the body shifting its molecular priorities to meet the demands of a different phase of life. All of it, every telomere, every repair enzyme, every circadian oscillation, was inherited. passed down through billions of generations of cells, from the first self-replicating molecules in warm, shallow water to the 100 trillion cells that make up your body tonight. And tonight, that same process continues. The delta waves are slow, the interstitial spaces are wide, the fluid is moving, the breaks are being sealed. You don't need to direct any of it. You don't need to understand it for it to work. You just need to rest. The cells know what to do. They've been doing it for a very long time.